Now we discuss the Vibro cholera. That is the second most important species, the Enterobacteriaceae. They are the part of the gram-negative rods and producing a severe life-threatening disease. Clinical manifestation is called Vibro cholera. Now, three strains, medically important virus, are observed: Vibro cholera, parahemolyticus, and melnificus. Now, the first one, that is the Vibro cholera, is the most important, medically important virus species in the Enterobacteriaceae. Morphologically, they are gram-negative rod, but curve-shaped, or you can say comma-shaped. And sometime they are observed in a single or sometime in pair. They are motile, as they containing a flagell. Transmission of this microorganism is by orophycal route, contaminated water, and the food. Now it is said that the Vibro cholera microorganism is required to produce pathogenesis near to 1 billion microorganisms ingested by the patient, and it colonizes the small intestine. So the pathogenesis start here. This microorganism produce two types of enzymes, oxidase and mucinase. Oxidase is the property of the gram-negative Enterobacteriaceae. Is produced by the in the majority of the Enterobacteria, while mucinase is produced by this Vibro. A single type of toxin. Remember to this name for the MCQs. Chlorogen. That is the Vibro cholera enterotoxin, and this is the major part of the toxin, which is the major parts of the virulence factor of this microorganism. Now, pathogenesis start after the ingestion of microorganism. Mucinase enzyme is responsible when microorganisms reach to the intestine, cross the stomach, they attach to the microvilli and the bush border, the intestine, epithelial. It releases the mucinase, and this mucinase enzyme is responsible to trigger the toxin chlorogen. Mean they stimulate the production of chlorogen toxin, which is released by the microorganism. And as the other toxins, this toxin also two parts. It's active and B is the binding. Binding part is help to the attachment, the intestinal epithelial cell, intestinal wall, and active is the active part. Now the A subunit, when inserted into the cell wall, the epithelial cell, they catalyze here ADP ribose, a type of protein present in the log form intestinal wall. They start to 1, which stimulate the adenyl cyclase. Now this enzyme increase the production of cyclic AMP. This cyclic AMP will increase in amount in the small intestine, so they do dual action on this intestinal wall. First, they stop the absorption of water, an important electrolyte, from the intestinal wall. Second, it increases the secretion of the water from the intestinal wall to the gut. I mean, remove the water from the body to this intestine, and a bulky watery diarrhea produce. During the diarrheal condition, the important salts as well as water is released from the patient, and a severe dehydration with the electrolyte imbalance, the lethal condition produced in the patient, which is within the few hours, and it leads to the lethal condition if properly not handling to the patient. In this case, if we see the clinical finding, so within few hours the pathogenesis start after the ingestion of the microorganism. Remember, the amount is much high. That is the reason this disease is not so much common in our vicinity, but sometime it is the outbreak of the cholera occurs. Few hours of the incubation period of this microorganism will start to the sudden onset of the nausea and vomiting and profuse diarrhea. Rice water stool, that's the sign. Sometimes they ask in the MCQs from you, rice water stool, green in color with foul smelling and profuse. Profuse mean it does not take an interval, but patient complained of passing the stool suddenly and repeatedly. So the complication starts, and the major complication is the dehydration, which results different type of metabolic disturbance like acidosis, shock, as the removal of the large amount of the water, and you are also one of the complication. And if the patient not handle suddenly and in emergency, treatment started by the antibiotics as well as by the IV line, the saline and the glucose drips. So the complication occurs and the death is the of the untreated patient we observe. Now in the lab diagnosis, the health worker do not waste the time by using lab diagnosis because this is a severe life-threatening position. So they start the IV line as well as antibiotic therapy. But to confirm, because the cholera is the highly infectious spreading disease, so to confirm whether this thing is the Vibro cholera or the diarrhea by other microorganism. So to confirm this, we put the procedures of the lab diagnosis. In the lab diagnosis, we perform again the stool microscopy. In stool microscopy, easily we can see a curve-shaped microorganism abundant in the stool. Second is the culturing. In the culturing, we use a selective media for this microorganism. Remember, the name is TCBS, thiosulfate citrate TCBS, thiosulfate citrate bile salt agar. This is the selective media for this microorganism. That's the Vibro cholera. That's the Vibro. We can use alkaline peptone water, and after the growth we can confirm this by gram staining, and by biochemical test we can confirm by a slight agglutination test this microorganism. Dear student, remember about the Vibro cholera in the examination, they do not ask. If they ask, so you can write from the microscopy, stool examination, and stool is the specimen. In the stool examination microscopy, and then go further for the culture. But the prophylaxis or the preventive measure asked again this microorganism, because this is the highly infectious and spreading disease, and due to the contamination of the majority cases of the contamination of the drinking water, because our sewage system in the cities, you can observe that the pipelines, they are passing from the sewage lining, and if this line are rusty or there is some pores or not the fitting is properly of these pipes, so this microorganism can be mixed with your drinking water and used to your tank as well as up to your glass, and this is ingested by you as well as the food. So isolation of the patient, proper disposal of the excreta, as well as proper treatment of the carriers, and the chemotherapy with antibiotics. The best one is the vibromycin and the IV line. You can write the improvement in the also the poor hygienic condition. So this is the prophylaxis or the preventive measure against this disease, that the improvement of the hygiene, sanitation, eat properly cooked food, and at the time of, if you heard about the cholera in your vicinity, so use the boiled water or the mineral water, not your drinking water from your pipeline. And the mass education means health education, that how the Vibro cholera is spread, as well as about the sanitation and properly washing of the hand of the mothers, and washing of the, if you handle the patient, your hand, as well as detection by the different test of the carriers and the chemotherapy. Thank you very much. This is the end of this topic.